weekend. This is your reminder that there is no school on Friday or Monday. It's a little confusing on the school calendar. If you were confused, on Monday it says that classes start at like 5:30 p.m. So if you're unlucky enough to have a class that starts at 6 p.m. on Monday, you do have to go to it. But the vast majority of people don't have classes that start that late. So most people will not have school on Friday or Monday. Because it is a scheduled break and it's built into the schedule, there's nothing for you to do. I don't have like extra recorded lectures or anything like that. It's just a good time to catch up if you're behind, maybe start working on those Connect assignments, but there is nothing specifically due for over the weekend. The only thing I will say is if you haven't yet watched it, make sure that you watch the recorded lecture I posted for Monday because the projector wasn't working and I had to cut lecture short. I posted a recording from last quarter. Sorry if the editing on it is weird. I had to splice together 2 separate videos. Hopefully it worked mostly, but there may be a little bit, my editing skills are not top notch. But yeah, if you haven't watched that yet, just make sure to catch up with that, watch it at some point. But yeah, so I won't see you guys again until next Wednesday. Right? Okay. All right, so we are picking up where we left off at the end of that video that I posted. So again, if you have not watched that video, you missed some stuff, go back and watch it. I'm not going to go over it again in class. I'm considering it covered. Is there something going on? Everybody's very whispery today. You have any questions or something? Okay. All right, so we are picking up with symbiosis. So we talked, again, in the video, so if you missed it, just go back and watch it. It's all in there. But we talked about growth requirements for bacteria, specifically temperature and then the gaseous composition of the atmosphere and pH. All very important critical factors in determining whether or not something is able to grow in a particular environment. And then the last little bit of this chapter is just a couple of kind of random definitions, a couple of random topics that we have to touch on. So something I want to talk about is something called symbiosis. Now symbiosis, by definition, the definition is written at the top there, is a long-term association between 2 organisms of a different species. Okay? Now notice that definition, usually when we use the word symbiosis or symbiotic, when we use that word just in everyday life, it's not a common word in everyday life, but a lot of times people associate symbiosis or symbiotic as something positive. And that is not the true definition. A symbiotic relationship is just a relationship. It is not positive. It does not imply positive, negative, or anything else. It just means a relationship. Any relationship between 2 organisms of different species. Now, we can further define a symbiotic relationship by whether that relationship is positive, negative, or neutral by giving it these other names. And so these ones at the bottom are different types of symbiotic relationships. So symbiosis is kind of the umbrella term that just means any kind of relationship. And then you can narrow it down further by saying that relationship is mutualistic, parasitic, or commensalistic. Okay, so mutualism is a type of relationship where both partners benefit. Okay? So mutualism, a mutualistic symbiotic relationship, or a mutualism relationship, is when where both parties in that relationship benefit from that interaction. The example up here is nitrogen-fixing bacteria that live inside of leguminous plants. There are bacteria that live in the roots of bean plants for the most part. And the bacteria live in there because the bean plant provides them with food. And the plant benefits because the bacteria produces nitrogen for them. Very necessary. A really good example in human health are going to be the E. coli bacteria inside the human GI tract. So you have lots of bacteria that live inside your body, but one of those bacteria is Escherichia coli, also known as E. coli. It lives inside of your gastrointestinal tract, and you may have heard of E. coli before. It does cause disease sometimes. There are certain types of food poisoning that are caused by E. coli, but a lot of times E. coli actually helps us pretty significantly. So E. coli is a bacteria that lives inside your gut, and it does a lot of things for you. It helps you digest your food. It produces vitamin K. That's a big one. So the E. coli produces vitamin K, which is an important vitamin for blood clotting. If you didn't have vitamin K, your blood would not stay in your body where it needs to be. So your E. coli produces your vitamin K. It helps you digest your food. It also helps protect you from bad bacteria. It does a lot of different things for you. The E. coli living inside your gut, for the most part, is an extremely beneficial symbiotic relationship for the human. Now, the E. coli also benefits in that relationship because the E. coli lives somewhere where it gets regularly fed, right? We are routinely eating food on a very regular schedule. The E. coli lives in a very protected environment. The inside of our GI tract doesn't change much. For the most part, it's a very stable, protected environment. There are not typically other pathogens or predators that live in there, and it is fed a stable diet of food as we consume it. So both parties in that relationship benefit. The E. coli gets a nice place to live, and we get lots of really important vitamins and other things. So that is a mutualistic relationship between humans and the E. coli that live inside them. A parasitic symbiotic relationship is one in which one partner in the relationship is harmed and one partner benefits. Okay, so one benefits off the other's harm. And a good example of this is the one that's written right here. A human and a protozoa, or a human and any pathogen, really. So any bacteria or protozoa or yeast or anything else that grows inside of a human and causes disease for us is going to be a parasitic relationship, right? The human in that relationship is being harmed. They are being infected by a disease that's probably causing some type of symptoms. It's causing some type of harm to their body. What type of harm that is going to depend on the bacteria or the protozoa or the helminth or whatever else it is. And then the organism that is living inside of us is benefiting because they're feeding off of our body. Okay, so they are using our body as a place to live, but in the process, we are being harmed in that process. Okay. Kind of a more specific example, I guess, of the same thing would be a human and a helminth, a parasitic helminth. You know, we've talked about before those 35-foot tapeworms, right? So those tapeworms that live inside the gastrointestinal tract of humans, it's kind of in direct contrast to the E. coli, because the E. coli live inside of our gut and they eat some of our food and they get nutrients from our body, but it's in a balanced way that it doesn't harm us. Helminths, tapeworms, and things like that live inside of our body eat our food to the point of harming us. If you have an established tapeworm, one of the symptoms of having an established tapeworm is being starving all the time and losing weight constantly, because the tapeworm is literally eating all of your food. Doesn't matter how much you eat, the tapeworm gets it first. Okay? So this tapeworm inside of you is stealing nutrients from you, the human is being harmed, and the tapeworm is happy as a clam, right? It is being helped in that situation. In commensalism, that is when one partner benefits, but the other partner is unaffected. So in this case, we have, the one that they have there is a bird and a tree. right? A bird sitting in a tree. The bird benefits from the protection of the tree. It's a good place to build its nest, all that good stuff. And the tree doesn't care. It doesn't do anything to the tree. Another example would be like those little, I think, what are they called, like remoras or remores? I don't remember their exact name, but the fish that swim along with whales. They like attach to the bottoms of whales and they eat their scraps and stuff like that. The whale doesn't care. The whale is, 300 times the size of the fish. It doesn't care that he has a little hanger on. But the fish benefits because it eats its remnants. Now, within a human body, commensalistic relationships are kind of hard to talk about. Because in reality, most of the bacteria that live in and on our human body affect us in some way, either positive or negative. There are some skin bacteria. There are some skin bacteria that live on humans that don't really provide any benefit or negative effects. They just live there. They like it, it's positive for them because they eat the stuff that our skin produces. A lot of bacteria feed off of the oils that our skin produces or the dead skin cells that we produce, things like that. But for us, they don't bother us. Now, kind of related to symbiosis is something called a biofilm. And we've talked about a biofilm before, but I just want to touch on it again. So a biofilm is a synergistic ecosystem of microorganisms that work together to provide advantages over individuals. So what I mean by a synergistic ecosystem is they basically work by like a little village, right? These are a bunch of individual bacteria that clump together and form a little village of bacteria where they provide each other nutrients, they help deal with waste products and things like that, right? One bacteria will produce a waste product for itself that another bacteria uses as food. And then the cycle continues, right? So it's a little group of different types of bacteria and other organisms that work together. And just like you see with humans, right? There's a reason that most humans don't live solitary lives, right? Most humans live in some type of community, even very, very tiny villages and stuff like that. We work together, or we survive better when we work together and share resources. The same goes for bacteria. They survive better when they share resources and work together. So we're going to talk about biofilms. The reason I'm covering it again is we're going to start talking about how we control bacterial growth. So how we use, pH and temperature requirements and oxygen requirements to control how bacteria grow. We're going to start covering that in a couple of chapters. And one of the things that we have to consider are biofilms. Because if a bacteria is capable of forming a biofilm, it is much more likely to resist being killed. Okay. So a biofilm is a large type of symbiotic relationship. All of the bacteria in the biofilms have some type of relationship. And again, typically the bacteria inside of these biofilms will survive better than individual ones. Okay, now we're just going to talk briefly about bacterial growth. This is two pictures of bacteria growing, kind of how we expect to see them visually. In liquid media, or liquids, you can often see bacteria because of cloudiness. And you may have even experienced this, like at your house, if you have a container of juice or something in your fridge that went bad and it went cloudy. Another example of this that you may or may not have experienced on your own is this is actually pictures of urine. So the one on the left is normal urine. Normal urine is typically cloudy, but if somebody has an infection in their bladder, their urine can become very, very cloudy because of the bacteria growing in it. If you're in lab, you can see this growing in tubes of broth. The bacteria grow in there, and as they grow, the tubes will become cloudy. Another way that we experience bacterial growth visually is by growing them on agar plates. Each one of those little dots, this is a type of agar, it's called a MacConkey agar, where the bacteria turn pink. But each one of those little dots, like this guy right here, this individual circle, this one here, and all of this stuff in between, these are colonies of bacteria. That is where bacteria has grown in the exact same spot over and over and over and over and over again until it became a visible clump. So if you put bacteria on agar plates, you may experience growth like that. Again, you may have experienced this in your own house if you've ever left. You won't necessarily see colonies like this, but if you've ever left like vegetables in a vegetable drawer for too long and they get real slimy and gross, that's bacteria a lot of the times. If you also, this is a common one that a lot of people have. If you go home today and look at your shower curtain liner. If your shower curtain liner is red, it's because you are growing colonies of Serratia marcescens in your shower. Very, very common. It grows at room temperature in moist environments. That's why it likes to grow on shower curtains. It's not a pathogenic organism most of the time. It's fine, but wash your curtain liner or buy a new one. And a lot of times it'll show up as like pink dots first, and that is colonies of Serratia growing on your shower curtain liner. All right, so how do bacteria in specific grow? And we've mentioned this word before, but this is the first, I think, official time we're talking about it, is that most bacteria replicate through binary fission. Binary fission is the opposite of fusion. Fusion means two things coming together. Binary fission means one thing becoming two. So binary fission, all that happens during binary fission is that one bacteria makes a replicate of its DNA. So it has one chromosome, it replicates it so that there's two, and then that bacteria splits in half. And so now we have two bacteria, where we used to have one. Okay, so one cell splits, oops. Oh my gosh. One bacteria that splits into two. And those two resulting bacteria are going to be genetically identical. This one cell up here is exactly the same as these two genetically. Okay, their chromosomes are exactly the same. There is no combination of genomes happening here, right? There's not an egg and a sperm that combine together to create a new organism. The resulting two bacteria are genetically identical. So we typically think of this word generation time. So generation time in general is the time it takes an organism, any organism, to undergo one round of replication. Okay, so basically a generation time is usually defined as, and this generation time is usually more general, it's talking about all organisms. It's how long it takes an organism to replace itself in a generation, right? One generation to create a new one. For example, the generation time, the average generation time in humans is approximately 25 years, right? So that's about the time that humans, on average, they go from birth to creating a new generation. Okay? In humans, it usually takes a relatively long time. In things like cats, right? Cats are able to sexually reproduce when they're about a year old, so a year to 2 years. So in cats, the generation time is usually if they're not fixed and all that stuff, it's usually about a year or two. Again, humans is 25 years. In most animals, and even most plants, generation time is usually a matter of months to years, right? Even plants don't grow overnight. Plants take usually a few months before they grow and sprout seeds of their own. And then the next ones grow and sprout seeds of their own. However, there's something weird that happens when you're talking about bacteria, because bacteria can reproduce themselves in a matter of minutes. So the time goes from months or years to minutes. So generation time isn't really a useful measure when we're looking at bacteria. Another problem with looking at generation time is you can't see it when bacteria, typically, you can't really see it when a bacteria has created a new generation, right? In a human, it's usually pretty obvious. Somebody is pregnant for almost a year, and then they have a baby exit their body, right? It's usually a very obvious, very visual thing. Even with plants, right? They grow up, they sprout seeds, the seeds fall, the seeds become new plants, right? That's a very visual thing that you can watch and record and look at. In bacteria, however, you can't. So in bacteria, instead of talking about generation time, we typically talk about doubling time. So it's the time it takes a bacteria to double. And this is just a more accurate way of talking about the same thing. It's just a more accurate way of measuring bacterial growth, because we look at how quickly they double their size. And doubling time in bacteria varies wildly. Okay, so some regular examples are things like E. coli. E. coli, under the best conditions, will double approximately every 20 minutes. Okay, so that's why, again, if you're in lab, you can put a tiny amount of E. coli on a plate and come back 24 hours later, and there's going to be billions of copies on a plate. Okay, this plate right here, this grew overnight, 24 hours. Okay, and that started off as not being able to see anything. Somebody drew a line down the center with a teeny tiny amount of bacteria, and 24 hours later, there were billions of copies smeared all over this plate. And that's because E. coli doubles itself every 20 minutes. So when you start out with one bacteria, 20 minutes later, there's 2. 20 minutes later, there's 4. 20 minutes later, there's 8, 16, right? On and on and on and on and on. They double every 20 minutes. The bacteria that causes tuberculosis, its doubling time is 2 weeks. So there's a reason that tuberculosis is a very long-lasting disease. Most people who have tuberculosis don't even know they have it for months or years, because that tuberculosis sits in their lungs and it very slowly divides and replicates every 2 weeks. There's a big difference between 20 minutes and 2 weeks. So, and there's bacteria that are everywhere in between. So when we're looking at the way bacteria grow, we can kind of look at the way a typical or an average bacteria will grow within a lab. So the key things that we're looking at here is this is an average or typical bacteria. Not all bacteria are going to follow this exact curve, but this is an average or typical version of what happens. And this is also within a closed system. So what I mean by that is it's a system where nutrients are not being added and waste products are not being taken away. This is usually within like a lab tube or a lab plate. This is what will happen if you put a tiny amount of bacteria in a tube of nutrients and let it grow. You don't add anything extra. You don't take away waste products, right? It's the equivalent of putting a human inside of a closet with a stack of food and seeing what happens, okay? So this growth curve that we're looking at, again, is a typical or average growth curve of most regular or typical bacteria. And it, again, takes place in a closed system. No nutrients being added, no waste products being removed. So there are generally 4 stages of growth that will happen within this closed system. And the first one right here, so this spot right on the end, right there, is inoculation. That is where somebody took a small bit of bacteria and put it in a tube. They inoculated the tube. They added bacteria to this closed system. Now what we're looking at on this y-axis, up and down, is the number of viable cells, which means cells that are still alive. The number of viable cells over the amount of time. Okay? So this first phase right here, called the lag phase, is what happens right after inoculation. And what's happening here is the bacteria are typically consuming nutrients, they are metabolizing food, they are getting ready to reproduce, but they are not reproducing yet. Okay, and that is why the line is flat, because the bacteria are not typically reproducing yet. They're not doubling. They're not creating new versions yet. They are consuming nutrients and they're getting ready to start doubling, but they're not there yet. They just got added to all these nutrients. They need to eat some food, power up, get ready before they can actually multiply. Okay, so that's what the lag phase is. It's where there's a lag in growth. The growth doesn't happen. And the lag phase, the length of the lag phase is going to depend on the type of bacteria, but typically it's a couple of hours. And then after a couple of hours, bacteria will start to enter this exponential growth phase. And this means in this phase, the bacteria are actively doubling. And there is very little cell death. No to little cell death. Because at this point in time, there are lots of nutrients. And no waste products. Lots of nutrients, no waste products. Again, this isn't an exact metaphor because humans are a lot different than bacteria, but it kind of works if you think about it. You know, if you put 2 people in a tiny room with a stack of food, the first couple of weeks, what are they going to be doing? If you put 2 people there, they're going to eat, and they're probably going to do some stuff, right? They're in the room by themselves, they got nothing else going on. They're probably going to try to reproduce. That's what they would do. That's what the bacteria are going to do. They're in a closed situation with lots of food. Their natural biological imperative is to reproduce themselves. So they're going to be actively dividing. They got lots and lots of food, tons of nutrients. They got fuel. They are not a lot of waste products yet. And so they're actively dividing, and very little are dying. This is their most happy place, right? They are in the best moods. They don't have moods, they're bacteria, but they're actively in the best place because they have plenty of food and no bad toxic waste products yet. Okay? The third phase is this stationary phase, and this is what happens after that exponential growth, that large growth phase. They will then reach a phase where the number of bacteria actually remains pretty constant, pretty even. And the reason for this is the bacteria are still actively dividing. but are also dying at equal rates. And the biggest reason for that is because food is decreasing, right? Remember, we're not adding food to this system. So they're starting to have gotten to the end of their food sources. The amount of food is decreasing. And there is also a buildup of waste. Okay. So in the process of metabolism in all organisms, we all produce waste products. In humans, we produce urine and feces. Bacteria also produce waste products. Okay, so in this stationary phase, they're a little less happy. They're running out of food, and they're starting to die because of the toxic waste products. Now, the amount of division and the amount of death are relatively equal at this point. That's why it remains stationary. That's why the level kind of remains the same. There are equal amounts of division and death. And then, as you might guess, in the death phase, pick a different color. In the death phase, stage 4, there is more death than division. There is a lack of nutrients and high levels of waste products. So that is the average curve or the average path that most bacteria are going to follow when you put them in a closed system. You're going to have the lag phase where they're kind of building up nutrients, building up energy, getting ready to start doubling. Then the exponential phase where they're actively doubling, they've got tons of food, very little death, everything is great. Stationary phase where things are starting to slow down, cells are starting to die because of the toxic waste products, food is starting to run out, but there still is some amount of division still happening. And then the death phase where the food is basically gone, the waste products are reaching really toxic levels, there's very little division, and mostly the cells are just dying. So something that's really important about this growth curve is that when we are studying bacteria, we need them to be in a very particular phase of this growth curve in order to get accurate results. And which phase do you, if you had to take a guess, which phase do you think that would be? Anybody got a guess? Okay, well, let me ask you this question. Are you going to do better on a test? If I gave you a test today, are you going to do better if you're well-fed, well-rested, happy, in a good mood? Or are you going to do better if you stayed up all night, you're starving, you had nothing but coffee for breakfast, and you feel like garbage? First one or second one? First one, right? So think about it again. Which stage do you think we'd want the bacteria to be in? 2, the exponential phase, because that is where they are doing the best. They have plenty of food, they're doubling, right? That is where results are going to be the most accurate. So when we are testing bacteria, we want them to be firmly in that exponential phase. When we are testing bacteria for identification, doing all those 5 I's to identify a bacteria growing inside of a person. Now, most bacteria, we want them kind of in the middle of that exponential growth phase. So most bacteria kind of hit the sweet spot of that exponential growth phase. We usually say between 12 and 18 hours after inoculation. About 12 to 18 hours after inoculation is when bacteria are typically in that kind of happy stage of the growth curve, that exponential growth phase. And that is why, again, if you're in a Monday, Wednesday lab, some poor grad student has to come in on Sunday and put bacteria in tubes for you to test on Monday. If you're in a Tuesday lab, somebody comes in on Monday, so the bacteria is ready by Tuesday. Okay, and that is also why when we test bacteria in the hospital, we inoculate it, and then we come back about 18 hours later and look at the results. Because we want to run tests when they're in their kind of happiest place. They have plenty of food, they're going to act their best. All right, and then these two slides are about how we figure out how much bacteria is in a sample, but I think because we've kind of fallen behind a little bit, we're actually just gonna skip these last two. Okay, so they won't be on your test. We'll skip that last little bit. Try to catch up a smidge. All right. Okay. So next we're going to move on to chapter 7. And what we're talking about in chapter 7 is metabolism. Now that's a word that most people are familiar with, right? You've heard of metabolism, usually in the context of like human metabolism. And we think about people having a high metabolism or a low metabolism and things like that. But what does metabolism actually mean? And that's what we're going to talk about. And how bacteria, in specific, metabolize things. So metabolism, at its most base definition, metabolism is the collection of chemical reactions They are the collection of chemical reactions that occur inside of a cell. So it is everything that a cell does in order to be a cell and in order to be alive. It's the production of certain types of macromolecules. It's the production of new cells. It's replication. It's digestion of food. All of the things that a cell has to do in order to be alive. And then another part of metabolism is kind of within that is how a cell utilizes energy. How a cell utilizes energy to make a reaction happen. So I hesitate to use the word make energy. We kind of think about it in terms of like making energy. But the reality of it is that in no part of metabolism is energy actually created. What's happening is you are utilizing and harvesting energy from the chemical compounds that you eat. You're not creating it, you're harvesting it. Energy is neither created nor destroyed, right? That's one of the basic foundations of thermodynamics. Energy is never created or destroyed. It gets redistributed and reharnessed, right? Even plants and things like that get energy from the sun, they're not creating energy from sun rays. They're harvesting electromagnetic radiation from sun rays and turning that into energy. So metabolism, again, is the collection of the chemical reactions that happen inside of a cell and how a cell utilizes energy to make those things happen. The most basic form of metabolism that humans can relate to is digestion. It's how we break down our food, the food that we eat, how our stomach breaks it down, absorbs it, makes energy out of it, and then uses it to power everything that we do. Right? How it powers our heart beating and our lungs moving and our muscles moving and reproduction and all of the things that our body is capable of doing is done because of digestion. Without food, our body would not be able to complete those things. And that collection of reactions is metabolism. Now, metabolism can be broken down into two general types of reactions: anabolism and catabolism. Anabolism is the building of large molecules from smaller building blocks. And then catabolism is the opposite. Catabolism is the breaking down of large molecules into smaller pieces. And we're going to look at in just a minute that metabolism is basically just catabolism and anabolism happening over and over and over and over again. We break stuff down to build it back up again. We break it down to create energy, which builds other things, right? It's just these two types of reactions circling in a cycle and happening over and over and over again. So again, anabolism is the building of something, and catabolism is the breaking down of something. Now, the key thing here is in order to build something, building larger molecules requires energy input. So anabolism reactions require energy. And catabolic reactions, catabolism, releases energy. So the breaking down of things is going to release the energy that was stored in those molecules. We'll talk about how that happens in just a second. But again, almost all of metabolism is just these types of reactions happening over and over again. You break something down to release energy and use that energy to build something else. And then we take that and break it back down to release that energy and build something else. It just happens over and over and over again. Okay, and this is kind of an overview of what metabolism is. So in a human body in specific, we have the kind of coupling or the collection of catabolistic and anabolistic reactions. When we eat food, so this is the food we eat, right, contains nutrients, specifically most of the time things like glucose, maybe fats, proteins, stuff like that, but some kind of nutrient from outside or from internal pathways. So when we eat food, if we eat glucose, what our body is going to do inside of a human body is it's going to break down that glucose. It's going to take that glucose molecule that we ate through our digestive tract and perform catabolism on it. It's going to break it down, which, again, catabolistic reactions yield or release energy. So this is how we harvest energy from the food we eat. We break it down, and that releases energy. Those catabolic reactions release energy. And all of these processes, you may have heard of the Krebs cycle or glycolysis. Those are common things that people talk about in high school biology. Fermentation is another way that happens. Yeast does that. That is how yeast produce energy. If you put them in with flour and water, yeast is going to consume the sugars in the flour and produce energy for themselves. It's how they grow. Coincidentally, it also produces carbon dioxide, which is a gas that we use to make bread. Okay? That's what sourdough bread is. It's a bunch of yeast and flour and water that you let ferment. That also happens in a lot of like kombucha, certain alcoholic beverages. The yeast growing in there is producing its own energy. So that's a catabolic reaction. And then your body is going to use that energy that was harvested to do what it needs to do. In this case, it's taking those molecules and producing amino acids and then producing proteins. Okay, and then assembling that into an entire cell. All of these processes are anabolistic or anabolism, which are going to use energy. And then enzymes are a very complicated subject that we are really only going to touch on. So we're not going to talk too much about enzymes. This is really, the next 2 slides are really all we're going to cover for enzymes. This is one of those things that if you got into a lot of the science that we're not talking about, it'd be really, really complicated. And I think that there is a lot of stuff on enzymes in your book that we're not covering. So again, your reminder that if we don't cover it in class, we're not talking about it. So if you read about it, I'm sure it's true, but we're just not talking about it. So an enzyme is something called a catalyst. And what a catalyst is it's a molecule that lowers the activation energy of a reaction. So like we just talked about, all of those anabolistic reactions, those ones where you're building new molecules, require some amount of energy to happen. It's called the activation energy. Okay, so the activation energy is the energy required to make a reaction happen. Okay, and every reaction that happens has some amount of energy that is required to make it happen. It's called the activation energy, the energy required to activate said reaction. Enzymes, or catalysts, are molecules that lower the amount of energy required. Okay? So if you look at this chart, without an enzyme, it requires this much energy. With an enzyme, it requires much less energy. So it makes these reactions easier to perform. It means that they require less energy to perform. And enzymes are protein catalysts. So there are molecules that are catalysts that are not proteins. Even things like heat is a catalyst. Certain types of pH and things like that can be catalysts. But an enzyme is a protein catalyst. It's a catalyst made out of a protein. And now for naming enzymes, the only thing that I want you to keep in mind is that if a molecule ends in the prefix ACE, A-S-E, it is most likely an enzyme. Okay, so one of the ones that we're going to talk about in a minute, the enzyme lactase, and a lot of times these enzymes are named after the reaction they catalyze. Okay, so lactase is an enzyme that helps in the breakdown of lactose, which is a sugar. Amylase is an enzyme that helps in the breakdown of the protein amylose, or the sugar amylose. Okay, so that is how enzymes are usually named. So if you see a protein around, and it has the suffix ase at the end, it's very likely an enzyme. Now, the way most enzymes work is they have something called a lock and key design, which means that enzymes are very specifically shaped to catalyze one reaction. Most enzymes catalyze one reaction because the way that catalysts work, or enzymes work, is this lock and key mechanism. The reaction that they're catalyzing, the substrates, which are the things that go into the reaction. So a substrate is what goes into reaction. And the products are what come out of a reaction. So substrates go in, products come out. The substrates and the products have a very specific shape that matches up with the enzyme. So it's a lock and a key. They have to match up to that specific shape. Okay? So like in this reaction down here, the substrates going in, the reaction that we want to happen is we want to take these two half circles, the pink half circle and the purple half circle, and we want to slam them together to make a whole circle. that is kind of a light purpley pink color. Okay, so that's the reaction. We're taking 1 and 2 and forcing them together to be a full circle. That's the reaction. The enzyme that catalyzes them fits like a lock and key those specific substrates. Okay, so enzymes typically only catalyze reactions for one set of substrates. However, it is important to note that enzymes themselves are multi-use, meaning that while they only catalyze one reaction, like you have lactase down here, catalyzing the breakdown of lactose, lactase is only going to catalyze that one reaction. It is only going to work with lactose. It is not going to fit any other substrates. One reaction. But that single enzyme can be used over and over and over again to catalyze the same reaction multiple times. So they are multi-use, right? Kind of like, I guess, I don't know what I'm thinking. Thinking of like a fork, I guess. Like forks are only good for eating certain types of foods. Like you're not going to eat soup with a fork, usually. But if you have a metal fork, it can be used over and over and over again. You can eat lots of the same meal with it, but it only catalyzes one specific reaction. And this is a big part of why we talked about, and we're going to talk about it more, but we talked about protein shape and the layers of protein structure. And one of the reasons that it's so important for proteins to maintain their exact designed structure is because if they don't, the lock and key mechanism no longer works. So a lot of genetic diseases that exist in humans and other organisms are an enzyme deficiency or a problem with an enzyme, where an enzyme is the wrong shape and it no longer catalyzes the reaction it's supposed to. Okay? So these are just 2 examples. I talked about these before. The enzyme here is amylase, and what it's breaking down is starch, also known as amylose. Okay, and that little enzyme gets in there. It has a lock and key mechanism, meaning that it fits exactly to the shape of the amylose or the starch, and it breaks it into pieces. So it is catalyzing the breakdown of starch. And amylase is produced in a lot of places. In our human body, it's produced in our mouths and in our stomach. So our bodies are capable of breaking down starches, like in potatoes and corn and other starchy foods. That breakdown starts to happen in your mouth. Another one that we're very familiar with is lactase. So in this picture, the enzyme is shaped like a pair of scissors. But lactase is an enzyme that breaks down the sugar lactose, which is a very common sugar, particularly in milk products. And the lactose, as it comes into our body, cannot be absorbed and used by our body until it's broken down. So you have to have this lactase enzyme in order to break down that lactose so that you can absorb it and use it as food. And lactase deficiency is a very common deficiency in humans. It's called lactose intolerance. And lactose intolerance is when you either have a genetic defect where your lactase enzyme is defective and you're lactose intolerant from birth. A lot of people actually have an acquired lactase deficiency as you age. So that's a fun thing to look forward to when you're about 40, 50-ish. Most of us are lactose intolerant. Just that you gotta switch to almond milk, okay? Most of us are lactose intolerant once you reach about 45 or so. And the reason for that should kind of make sense if you think about how we develop, right? When you're a newborn, your body is specifically designed to consume milk. It's your primary food source. So it makes sense that when you're very young, your body would produce lots and lots of lactase to break down all that lactose that you're eating. Naturally speaking, milking other animals for their dairy products is kind of weird when you think about it. Like, I like cheese as much as the next gal, but if you think about it real hard, that we're milking other animals and consuming the milk meant for their newborns, it's a little weird, right? Like I said, I do it, I like it, but it's kind of weird to think about it. But as adults, we're not naturally designed to consume milk products, right? As adults, we're meant to be eating meat and grains, vegetables, right? We're not really typically supposed to eat dairy products. So it kind of makes sense that as you age, you would naturally produce less lactase. All right, so now that we've talked about enzymes, we're going to talk about how bacteria or cells in general find and use energy. So in order to carry out metabolic processes like building or breaking down things, our cells need a constant stream of energy input. But how and where they get that energy varies wildly. Partially based on what we talked about last time, right? Is an organism phototrophic or is it heterotrophic, things like that, chemotrophic? So it can vary wildly. Some organisms, like humans, mostly get our energy from sugars, fats, and proteins. Some organisms, like plants, get their energy from light. Some organisms, some bacteria get energy from like heavy metals. They can pull energy out of selenium and other weird types of metals. Some of them pull energy out of nitrogen, which is another type of element that's not very common. However, everything that we are going to discuss in this class, again, because we are focused on pathogenic bacteria. These are bacteria that like to live inside of a human. That is why we're talking about them. Most organisms that we will talk about get their energy from sugars. Okay? And most of the energy cycle is based on that molecule ATP. We're going to talk more about it in detail in just a little while, but that's kind of what that overview is showing you there. So again, like we were talking about earlier, energy is not created or made in cells. It's harvested. Okay, cells extract energy from nutrients and use it to fuel other reactions. Okay, so energy is never created. It's harvested or extracted. And so there are two types of general energy reactions, okay? There are the reactions that require energy and the reactions that release energy. So remember that those words, I know we've talked about exo and endo before. Exo, like an exoskeleton, right? An exoskeleton is a skeleton on the outside, like of a beetle. Exo means out of, or it released something into the outside. So an exergonic reaction, exo, is a reaction that releases energy to the outside. Remember again, like an exoskeleton is a skeleton on the outside. And then an endergonic reaction, endo, I just think N sounds like in, right? N in endo means that energy was brought in. So endergonic reactions are reactions that require energy. And then there's just a graph that kind of shows exactly what I was talking about, that exergonic reactions release energy, that the reactants have more stored energy than the products. So there's these reactants, what we have at the beginning, also called the substrates, have lots of stored energy in them. And as we break them apart, they will release energy. And then endergonic reactions are exactly the opposite. The reactants have less energy than the products. So to get there, we have to add energy to get there. Okay. And again, it's important to remember that energy is not created or made. Energy is harvested. One way of thinking about it is like a solar panel. A solar panel on your house does not make energy. It harvests energy from the sun. A windmill does not create energy. It harvests the kinetic energy from the wind. Okay. So how cells exchange energy is through this molecule called ATP. ATP, a good way of thinking about it is it's metabolic money. So energy is stored within ATP, and then it acts a lot like money because it can be exchanged, spent, traded, moved, saved, right? All of the things that you could talk about money, ATP can do the same thing. It's how your body stores and uses energy. And the way that it works is that ATP has what's called 2 high energy phosphate bonds. Now the reason that they are high energy is because we talked about before that adenosine triphosphate, or ATP, is an adenosine, which is a molecule like adenine, which is what you find in DNA. Knowing the exact structure of adenosine is not important, but adenosine triphosphate is a molecule with an adenosine and then 3 phosphates, triphosphate. The key to this is that these phosphates are negatively charged. Okay? All of these phosphates are negatively charged. Now, the reason that matters and the reason that we are able to store energy in this molecule is because think about what happens when you put charges together. I feel like this is something that almost everybody learned in high school. So when you put 2 of the same charge next to each other, what happens? Are they going to be attracted to or repel each other? They repel each other. Same charges repel. Opposite charges attract. Opposites attract, alike repel. It's like, if anybody has ever done this, right? I think most of us did it as kids. If you take 2 magnets, you know that a magnet has a positive side and a negative side, right? It's called the polar side of each magnet. Positive side and negative side. If you take the negative side of 2 magnets, they're going to repel each other, right? They're going to be really hard to push together, okay? And that force, that repelling, is energy, okay? That is a harvesting of energy. So I want you to think about those 2 magnets. If I had 2 big giant magnets in my hand that had the same charge, they were both negatively charged, and I tried to force them together, right? I could force them together, but what I'm doing is I'm using energy in my body, in my arms, to force them together, right? And I could hold them like that. I could hold them together in my hands, but that is requiring energy at all times. What will happen the second I let go of them? What will they do to each other? They're going to push apart, right? Like if you put 2 magnets next to each other and you push them together, when you let go, they're going to force each other apart. That forcing apart is energy. That energy is stored in the physical holding together of those 2 magnets. And so the way ATP works is these phosphate groups are all negatively charged. So what our body does to create ATP, or what any cell does to create ATP, is they basically use energy to force those two magnets together, right? Those two negatively charged phosphate groups that do not want to be next to each other. They force them together and they chain them together with a bond. They bond them together. And then the minute that bond breaks, the energy is released, like the magnets being forced apart. The energy comes back out. So the high energy bonds are the bonds between two negatively charged phosphates. Now, the thing that's maybe a tad confusing is that ATP has 3 phosphates, right? ATP, adenosine triphosphate, has 3 phosphates. But you only have a high energy bond between 2 phosphate groups. So this guy on the end, on this side, he doesn't have a phosphate. So this bond right here is just regular. That's not high energy because he's not being forced against another phosphate on that side. But these two right here, between these 2 phosphates and these 2 phosphates, are both high energy bonds. Okay? So basically when a phosphate group is forced onto this adenosine molecule, it requires energy, and that energy is stored in that bond. It's called potential energy because it's being held and stored in that bond. And when that bond is broken, it will release the energy again. The magnets will fly apart because you release them. Okay, so we talk about this. Adenosine triphosphate is ATP. That's an adenosine when you have 3 phosphate molecules. Adenosine triphosphate. ADP is adenosine diphosphate. It's when you have 2 phosphates attached. So this product, or this process here, of taking an adenosine diphosphate with 2 phosphate groups and attaching an extra phosphate to produce ATP, does that require or release energy? Forcing another phosphate on there. It requires energy. So if it requires energy, is that an endergonic or an exergonic reaction? Endergonic. N-in. Requires means it needs to come in. So this is an endergonic reaction. This also happens to be another type of reaction because we are building a bigger molecule. So is that an anabolistic or a catabolistic reaction? You're building something bigger. Anabolistic. So this is also anabolism or anabolistic. Those just mean the same thing. Okay. On the other hand, taking an adenosine triphosphate and breaking one of those bonds, releasing the bond to produce adenosine diphosphate and an extra phosphate that you just cut off, does that require energy or release energy? It releases it. Okay, it cuts that bond and the magnets are going to be forced apart. It releases energy. Is that endergonic or exergonic? Exergonic. And it also happens to be catabolic. Catabolic. Catabolism. That's not how you spell that. I couldn't figure out why that looked weird. Okay. Catabolic. There we go. Catabolic or catabolism. I'm checked out for the long weekend already too. All right. So that is how ATP basically works as metabolic money or energy money because these ATPs are created. We force the phosphate onto that ADP to create ATP that is the energy storage And then that ATP can move anywhere. It can go anywhere else in the body. It can go to another cell. It can be set aside for later. It can move to a different cell type. It can do whatever it wants. And then when the cell needs it, it'll break off one of those phosphates and release the energy and use it for something else. Okay. And this is just another picture of that ADP cycle. So it's exactly what we just talked about. But this up here is a triphosphate. So this is ATP up here. And this is a diphosphate. So this is ADP. But when you take ATP and break off one of those phosphates, you are going to release energy and phosphate to create ADP, but that energy is going to be released. And then on the other side, it requires energy to force another phosphate group onto that ADP to produce ATP. Okay, so it's just what we talked about over here, but just in a kind of showing it in a cycle. And then to add even more to that, a big part of how our body does metabolism is through what's called coupled reactions. So you take this ATP, ADP cycle, like we saw over here, and you use that energy for something else. So again, remember that glucose, this is kind of what happens inside of our body. We eat food, like glucose or whatever else our food source is, and our body breaks that down. Glucose breakdown, things like glycolysis and the Krebs cycle, is going to produce energy, okay? This is an exergonic reaction. It is going to produce energy. With the energy that glucose reaction produces, we are going to convert ADP to ATP, because remember, that is an endergonic reaction. It requires energy input. So the energy we harvested from the breakdown of glucose is going to be used to make ATP. And then this ATP will go wherever it needs to go. It may move to a different cell, it may be stored for a while, but then when our body needs to do something else, in this case here, we want to build a protein out of amino acids. Well, our body is going to break down the ATP back into ADP, which is an exergonic reaction, and will release energy. And it will use that energy released to build a protein, which is an endergonic reaction. Okay? And this is what happens over and over and over and over and over again. We break things down to release energy to build up other things, which will then be broken down to build other things. Okay? It just happens in a cycle over and over and over again. And that is the basis of metabolism, those coupled endergonic, exergonic reactions. All right. All right, happy Easter, everybody. I'll see you next Wednesday.